Okay, so good evening. Welcome to today's session. Today we'll be continuing our discussion from the previous class where we had introduced the concept of chip-seq data. Let me just switch on my video for a second. So we had initiated our discussion on chip-seq data analysis and the key steps that are involved in it and what are the types of tools that you would possibly need to analyze such data. And before we proceed with the discussion for today, I will quickly recap what we already wanted to. Just give me a second, I think. I will go back to the previous class so that we can resume our discussion after recapitulating what we had started off in the previous class. So, so we had introduced chip sequencing technique, and just to inform everybody, we will be having an examination of the topics that have been covered since the first exam. So if you remember, we had covered gene structure, function regulations, mutations, polymorphisms, and functional genomics, and introduction to sequencing methods up to Sanger sequencing in the previous class. Um, we will not resume, we will in the next test, we will carry forward our discussion. And also we will have a brief exam, pretty much similar to how we did in the last exam as well, where we will discuss the steps that are involved in analysis of sequencing data, high throughput sequencing data, what are the kind of tools that are required for the same, um, what are the parameters that need to be kept in mind when we do that sort of analysis. Next, we will also cover epigenetics in that concept, and that it will be a more elaborate exam because the number of topics in the first one were much lesser. But in this one, we will cover a couple more discussions. So I will go over I will recap some of the topics today, and I will also carry forward a little more of the discussion in the next class, and then we'll have the exam. So we'll have the discussion pretty soon, don't worry about it. And also the next class will be on Friday. So taking just a quick look at what we have already discussed. So we had started off with a discussion on chip sequencing method. So if you remember, chip or chromatin immunoprecipitation is a strategy that is used to study DNA-protein interactions. Largely, the types of proteins that can be studied extensively using this method are transcription factors, chromatin remodelers, basically any protein that possesses a DNA binding domain and interacts with DNA using a certain portion of the entire protein. So it is an extensively used strategy in epigenomic research and is invaluable in that context. Largely, what you would find as a commonality in these methods is that there is one protein, which is your target protein of interest. You have the genome or the DNA isolated from whatever cell system you are interested in studying. Most commonly, that is it is going to be a model or a cell line model, which you can use for extensive analysis. And you need to have an antibody, which is very specific to your protein, not just specific. It is very high grade. When we say high grade, it basically means the antibody has undergone extensive purification, as a result of which, when you use it for these sort of experiments, which are very costly and very what do I how do I put it? Like, which depend a lot on the purity of the reagents that you use in such context, which are very sensitive. Let's put it that way. In that case, the purity of your antibody really matters, and that is something that needs to be taken care of. And also, one more thing that should be kept in mind whenever you are planning a chip experiment is that your antibody should be able to recognize its epitope when the DNA binding protein is bound to DNA. In that state, it should be able to recognize your protein of interest. Otherwise, you will end up getting false results. So that is pretty much the idea. Now, apart from that, when we do chip qPCR, which is what I had discussed in a couple of classes ago, then you are specifically looking at some XYZ gene, which you are hypothesizing that your protein of interest is able to regulate or is able to bind to. So, for example, if gene X is regulated by protein Y, you would immunoprecipitate protein Y from your cells and then check whether they are able to regulate gene X by using primers against gene X, right? So while that is all well and good, it's not always going to give you a complete picture, and sometimes your results might not be very conclusive. But if your idea is to look at what I mean to say is that chip qPCR is more of a validation of the results that you expect to see using other strategies. It's not solely all you need. Chip-seq analysis or chip sequencing, as any high throughput strategy for that matter will be able to do, is give you a more comprehensive picture, give you a more detailed idea of what is appearing inside the cell without any other bias, and it will also give you a more um, consolidated image of what happens in a cell in a certain condition. For example, if you're doing a treatment or some sort of a genetic modification in your cells, then using chip sequencing is going to give you a picture of all of the changes that occur as a result of that response, and the interplay of different proteins and different regulatory pathways can also be inferred from such a project. Now, naturally, one might ask, why don't we always go for chip sequencing, and why do we at all need to do chip qPCR? The short answer is money, because it costs a lot to go for chip sequencing. Secondly, it is, while very useful, any sequencing, high throughput sequencing method is always subject to errors. And to bypass those errors, it's always a better idea to validate it at a small scale. And the most commonly used validation in that case is using chip qPCR, like I just mentioned. Now, what all these challenges and all these shortcomings as well as advantages, they sort of balance out each other. And eventually, what we understand is that the role of a chip-seq is indispensable. But in today's day and time, there are a lot more informative techniques which are more elaborate, more expensive, but at the same time, they give you much more detailed information about higher order protein, DNA protein interactions and higher order chromatin architecture. And they are also extremely informative and extremely complicated techniques as well. However, ChIP-seq still remains regularly used in any research publication that is focused in this domain. And so it's always a good idea to learn about it and know how to utilize publicly available data for our own use. So, or just a quick recap, if you look at this example, here you have your representative chromatin onto which your protein of interest is bound. In this case, that protein of interest is p53, and you use an antibody which is able to bind to p53 and is able to recognize some motif on it. And what you want is to sort of shear your chromatin so as to generate smaller fragments which take up majorly the nucleosome and a couple of bases extending from the nucleosome, which shows you that this is the site where the protein is expected to bind. And this is what your antibody gives you, and then you discard the unbound fragments and you sequence the purified fragment. That gives you this sort of a map, as you can see, which is sort of a track for all of the regions where DNA has been sequenced from after it has been aligned to the reference human genome, so that we can conclude which regions are bound by our protein of interest inside the cell under the given condition that we are studying. Now, these are the different types of histone marks, like I had already mentioned in previous class, where you have histone modifications being a very crucial sort of sub-branch in context of chip sequencing and analyzing chip-seq data. So you have H3K4 monomethylation, H3K27 acetylation, H3K4 trimethylation, H3K36 trimethylation, H3K27 trimethylation, and H3K9 trimethylation. These are the most common core histone marks, and they are very each of these types of modifications and the associated data which is obtained from sequencing cells which are possessing that modification at a given point of time. That information can give you a lot of different types of conclusions based on which modification you are studying. For example, H3K27 acetylation is going to tell you, let me just switch this off, it's going to tell you whether it is associated, it's going to tell you whether a certain region is transcriptionally switched on and is being driven by enhancer regions as well. Yeah. H3K for ME3 is going to tell you whether it's bound to a promoter region. Then you have H3K36 ME3 for transcribed, actively transcribed regions, H3K27 ME3 for polychrome group proteins driven repression, and H3K9 ME3, which is associated with heterochromatinization for certain region, which is again shutting down of transcription. So the distribution of histone modifications across transcriptional regulatory elements can be seen here. This is an example in T cells. This is what you can mostly be able, what you'll be able to see. So if you observe here, you'll see there's a contrast between the spread of these different modifications in each case. So in context of an active gene, you will see H3K4 MA3, which is associated with promoter regions, for that modification to be present in the near the promoter regions of the enhancer, etc., in an active gene, whereas it is absent in a silent gene. Yeah. Not absent, but quite reduced. H3K9, 14AC, etc. And you can see the different sort of modifications which can be seen here. Now, the distribution of histone modification is quite variable depending on what is the condition that you are studying and what type of genes you are studying at that point of time. Now, characterization of chromatin signatures in immune cells have mainly been performed in T cells, and these distributions are from T cell ChIP-seq data. Enhancers typically they can be characterized by the presence of H3 acetylation, the methylation, H3K9ME1 T cells, and T cells, Ipg islets, etc. Yeah. So again, the key steps are as follows. The initial steps are similar to what we had discussed for ChIP-qPCR. First, you have cross-linking and fragmentation, whereby the DNA bound or DNA binding protein is first cross-linked to DNA within cells using formaldehyde, which is able to strengthen the interaction between protein and DNA. Then you lyse the cells, and the fragmented complex can be divided into small pieces by sonicating or by enzymatic digestion. Next, MNase digestion is often preferred over sonications because that helps to fragment the chromatin and it can trim the link DNA, providing more accurate mapping. So MNase digestion is often also used. Again, this is something we previously discussed. Then you do the actual precipitation step, whereby specific antibodies against your protein of interest and binding to the epitope which is exposed, they are able to bind. Then they form complexes which can get captured on resin, and it can be washed off. DNA cross-linking with the protein can be reversed because you do not want very strong intensity of binding, which will eventually hamper your DNA elution, which is the actual target molecule that undergoes sequencing. The proteins and the RNA can be digested, and effectively what you purify is your DNA molecule, which represents the sequences bound by the protein and is then ready for sequencing. Next, the sequencing library construction. End repair is performed on the purified DNA and sequencing adapters attached to the DNA fragments. Then they can be amplified using PCR. The amplified DNA is size selected using gel electrophoresis, and that creates a sequencing library. Again, these are mostly taken care of commercially, but learning basics is always a good idea. Then you have the actual sequencing step, whereby the prepared sequencing library can then be sequenced using one of the available NGS platforms like Illumina, Solid, or Helicose. The platforms involve different methods of amplification and sequencing by synthesis, allowing the massively parallel sequencing of many DNA fragments. Next, this is a comparison of the ChIP-seq method with another strategy, chip-on-chip, which we discussed in the previous class. And the chip-on-chip actually, it sort of blends together it blends microarray and ChIP-seq somewhere in the middle. So chip-on-chip uses DNA hybridization with sequence-specific probes on microarray chips, and ChIP-seq uses massively parallel sequencing or NGS technology. Then you have genome coverage. In case of chip-on-chip, very limited coverage is available on the array because only very discrete specific regions of the genome can be analyzed. ChIP-seq allows for comprehensive coverage, including repetitive regions that might be missed by chip-on-chip. More cost effectiveness is found in chip-on-chip, whereas ChIP-seq is more expensive due to advanced sequencing technology. Then you have better resolution in ChIP-seq as well. It can give you sometimes up to single nucleotide resolution and provides precise mapping of DNA binding sites. However, resolution is quite limited when we talk about the chip-on-chip since it is a microarray-based method. And the amount of starting DNA needs to be much higher when we talk about chip-on-chip, whereas relatively less DNA can serve the purpose in ChIP-seq technology. Yeah. So I was just quickly revising last class's topic before moving forward. So so this shows you a sort of flowchart of how chip-on-chip needs to be performed. So this is an example where from wild-type embryos, DNA is isolated, formaldehyde fixation is performed so as to cross-link the DNA and our proteins of interest, and that is covered under the purview of preparation of chromatin. Then immunoprecipitation is performed by adding the targeting antibody. Then after IP, you cross-link, you reverse the cross-link so that DNA and protein interactions are no longer favored. And you amplify the isolated DNA, clenolabeling is done, and then the labeled genomic DNA, which has a different color label, both of them are allowed to bind. And what you would expect is that wherever hybridization has occurred, the fluorescence emission is supposed to be of a different wavelength, which is going to tell you that there is this that hybridization has occurred. And then you statistically analyze it, visualize it, integrate data to finally interpret your results. So the question that we addressed in the last class, what exactly can you sequence or what is preferably sequenced? You have your input material that most certainly has to be sequenced whenever you are carrying out ChIP-seq data analysis or ChIP-seq experiment overall. Why? Because in ChIP-seq, the results that you get finally are relative to the control or relative to the input. Basically what is happening in a cell at a basal level versus when I enrich a particular protein by immunoprecipitating it, what changes do I observe in that case? So therefore no measure is absolute. These are all discrete measures. Then what you end up with is sort of something like this, which you have to interpret and decode, and then finally figure out whether a certain DNA protein interaction is occurring or not. Then you have these representative examples of the different types of enrichment. You have single points, which are very, very sharp, very neat peaks, which define that there is the binding of a certain transcription factor in a certain locus or some histone mark which is present. Then you have broader regions, um, mostly some histone marks and uh, polymerase enzyme, etc. And the type of the peak or the type of enrichment you will get also depends on the nature or the physiological function of a particular protein. If a certain protein is very is sort of a chromatin remodeler, then its binding is expected to be much more wide because it can bind over a stretch of DNA, and it's not the exact sequence of DNA that always makes a difference, but instead it's the function of the protein that makes all the difference. And third is virtually everywhere, where you see peaks everywhere along the entire gene. So we had initiated our discussion on gypsy data analysis steps. So first is FastQ, pretty much similar to what we saw for RNA-seq as well. So you have to initially sort of process your reads to make sure that there are no errors, no contamination, and you can get rid of any low quality reads, and you can get reads of a certain length, etc., etc. Then you have to align it to the genome. Then you have to filter BAM files. You have to do yeah, you have to filter the BAM files, do peak calling, assess the quality of your chip experiment, and then compare the difference between the binding or the enrichment of a certain protein between your control group and your experimental group. And then you use that for annotation, for functional enrichment, for motive discovery, visualization, etc. So yeah, the reliability of chip analysis governs is largely governed or controlled by antibody quality. So what you want is you want more and more enriched signal for your protein of interest, and you want the most possibly reduced signal to noise ratio in an ideal situation. Now, false positives and false negatives, they exist everywhere. They're derived from non-specific antibody DNA binding, might compound the analysis, and they should be, any result you get should have multiple validations with many different antibodies. That's always a good practice, while not always feasible. So, yeah, these are the different types of peaks that you can possibly get. So you can have either very neat, very distinct peaks, like you can see for point sources. Then for certain modifications like HTK27ME3, etc., you can see that these peaks are not very well defined. Instead, they sort of spread out like a mat, and that indicates the type or the function of that protein. Third, you have big sources where you can have both these sort of attributes, and yeah, that is pretty much it that we discussed in the last class, I think. Yeah, just these two slides. One additional option that you always have is to either have a single end or a paired end read. So yeah, so single end reads, they are able to sequence the DNA fragment from one end, making it suitable for shorter fragments. Paired end reads, however, can do so from both ends, which makes it very, the utility of it much more enhanced. And yeah, so you can have different ranges of your sequencing read. You can specify that based on your requirement. Um, then you have targets of RNA-Pol2, which are abundantly present in many genomic regions or localized histone modifications, such as H3K4, ME3, and you should always use negative controls during your IP experiment and input controls so that you can plot a relative comparison between input versus your IP sample. Finally, among the steps in ChIP-seq data analysis, the first two steps are pretty much similar to what we had already observed in our previous high-throughput sequencing data analysis. So you have your QC of your raw sequence that has to undergo a check. Then you have all of those different parameters for ensuring that data is pre-processed. And then you have a QC of a raw sequence. And if it has adapter contamination, then that is different. That can be taken care of by Trimgalore, who made Trimomatic, I think, who is able to, which is able to detect any such adapters which might be present so that it can clean them off. And it does not hamper the actual, it's not necessary, let's just put it that way. And you also have QC of the raw sequence composition so that you can get an idea that distribution of all of the bases are equally probable in each case. Yeah, so that was our discussion for the previous class. So I'll take a quick 2 minutes, not even that, 1 minute break, and I'll be back. If there are any doubts, you can let me know, and then we'll move forward for today's class. So I'll just be back. Okay, so let's continue. So I'll move ahead with detailed steps in gypsy data analysis. If you have any doubts at any point, please let me know. We will move ahead with that first. And apart from that, this part, if you joined the previous class, you would already have noticed that we discussed this, how you can access, how you can download and proceed with publicly available gypsy data, where to get it, and what are the different parameters you need to adjust in order to make sure that what you're getting has a matched input, that it has replicates, what is the platform that they have used, just in case it's something you want to validate for yourself. All these parameters should also be taken care of because that usually helps. So, and this part is what we'll introduce, the 3D chromatin architecture part. But yes, that needs to be taken care of in more detail, which I'll do in the next, probably the next class. And also, before proceeding forward, because this will probably be the last module we'll be discussing. So before we move ahead with that, in the next class, we'll have a brief exam once again. I hope more people join. If not, the link will be available, will be shared to you over email. But I would prefer if people do it live. But irrespective, we will have a discussion on the questions that I will frame for you people. The topics I'll let you know. Let's just, if I look here, basically we had discussed till this initial portion on Sanger sequencing, but it won't be very elaborate. Don't worry, you don't need a lot of additional prep. But if you have attended classes and if you can just quickly brush up the topics with me, that'll help you. I mean, that'll be good enough in my opinion. Uh, what I was thinking we will be covering is this initial part on NGS, unraveling genome, exome, and transcriptome, because these were kind of overlapping topics. And then analysis of high throughput sequencing data. What are the commonly used in silico tools? So we'll be doing that. And 2 or maybe 3 classes later, we will, like after I've covered another portion, I'll tell you that also in real time. We will look at DNA methylation associated topics so that we can cover those in more detail. And hopefully, once we are done with all of the topics that I wanted to cover for this course, we will finally have sort of the last test that has all of these remaining topics. Don't worry, we won't have a complete, I don't think I've not planned anything of that sort that will have a complete test because these topics are quite vast, quite far and wide. So I doubt it'll be possible for anybody to brush up on all of these topics. But just to make sure that you have a few questions on each topic and you know how to tackle these sort of questions if you come across them, I am conducting like tests periodically so that it helps you better, I think. So that's the plan. But anyway, moving ahead, initially what we had discussed that if you get those raw reads, if you remember, we I had shown you how to retrieve these publicly available data, and then also you saw that you can get the fastq.gz file from SRA or from NCBI directly as well. So that part you can download. You can directly carry out the pre-processing steps with fastqc and your Trivomatic to get rid of adapters, get rid of low quality reads, remove any biases, trim sequences, etc. We have already discussed all of this in our initial discussions on high throughput sequencing data analysis. So I'll not go over all of that detail again, but a lot of the steps you might see are quite similar to what you saw for whole exome sequencing or RNA-seq data, just that the eventual interpretation of results is done differently, but the first few steps of data processing are quite similar. So after the initial trimming and the uh, quality check has been performed, the next step is read mapping. Now, what do we mean by read mapping? It basically kind of means alignment. If you remember, we had discussed previously HiSat2, STAR, all these algorithms. And in addition, for ChIP-seq, what is most commonly used is the bow tie or bow tie 2. Most commonly, bow tie 2 is used in ChIP-seq data analysis. So basically, you will have your raw data, which can which can be mapped using many different types of tools like Bowtie 2, BWA, Burroughs Wheeler alignment or algorithm. I cannot recollect exactly, but it's also a different strategy. So these can consider indels, which are insertions and deletions by gapped alignments, which is appropriate for long or paired reads. And there are several in output formats as well for the map files like SAM, BAM, CRAM, and tag-align. When the BAM format is most commonly used, you will also have CRAM format. So again, if you remember, we had discussed that after alignment, you get a SAM file, which is quite a large file and makes it very difficult to perform analysis. So usually you convert that into a BAM file using a tool known as SAM tools, if you remember. Again, once we brush up the technique, the high throughput sequencing analysis part, you will sort of recollect all of it. So BAM format is most commonly used, but you also have a CRAM format, which is also under consideration. Then the reads which have mapped to the same genomic location or position get filtered as redundant reads, and the remaining non-redundant reads are utilized for the analysis part. For BWA, the mapping rates are much higher. Uh, Yeah. So if you see here, quality control is done by FastQC, which gives you your FastQ file, which is aligned to the genome using Bowtie 2-like algorithms, where it preferably only keeps the uniquely mapped reads and discards, like again, that's a parameter you can adjust yourself, but you can discard all of the non-aligned reads, or you can keep them for some sort of future analysis. Then the BAM undergoes filtration and then peak calling, and finally bed file analysis is carried out. So for BWA, the rate of mapping is usually higher with an equally similar increase in the number of duplicate mappings identified. Post filtering, you get a significantly higher number of mapped reads, and which gives you much larger number of peaks being called. When we compare the peak calls that have been generated from two different alignment algorithms, BWA peak calls become a superset of those which have been called from Bowtie 2 alignments, and it also requires further study. So what did we mean by this entire idea? The entire idea says that basically for different algorithms, there are different sort of standards or different default settings which are in place. So whenever you are using multiple tools for the to draw conclusions, then at that time it's a good idea to to sort of normalize them against each other. And yeah, that's pretty much it. But what you should ideally do is that you should have a complete experimental set. For example, if you again look at any publicly available database, then in that you will have all the experimental information available. So whenever you proceed with the analysis, which is an elaborate step, you should make sure that your complete experiment or complete project has uh, multiple samples which can be used for normalization, and between two different projects or between two different sequencing machines or softwares, there can be a lot of difference and alignment might not be very reliable. The comparison of the alignments might not be very reliable. So it is a good practice to do what I mentioned before. So next, what happens till now? What do we do? What did we do? We just aligned our fragments that we have eluted post immunoprecipitation with the genome. Next step is peak calling. So what do we mean by that? The peak calling step. Let me just. The peak calling step identifies significantly enriched loci in the genome. So what do we mean by that? We mean that we till this step right before this, what we saw was that there is the alignment of the reads that we have obtained from DNA bound regions. They are located from which region of the genome? Only that part is known. Now, what peak calling is going to do is going to identify significantly enriched loci. It's not going to look at only the gene or a certain position or anything of that sort. It is going to do a global analysis and is going to give you all of the significantly enriched loci in the genome. So generally, these are returned in the bed format. That is also just a simple file format .ved. And normally, what you will do is you will analyze that file. You'll use another tool called as max2 to determine whether it is a statistically significant difference between the input and your immunoprecipitation condition. So although ChIP-seq peaks do not have strand information, it can be estimated from the gene information when focusing on histone marks that are enriched around the transcription start site. And for peak calling, Max2 is most commonly used. It also gives you the stats for it, like I mentioned. However, you cannot at any point rely on any technique as being 100% accurate. A better idea is to obtain a large number of peaks with a relaxed threshold that contains both true positives as well as noise. And from there, you sort of extract subgroups using another way to improve specificity. And one example of that is selecting consistent signal among biological replicates using irreproducible discovery rate or IDR. So now, yeah, so if you look at the image on the right, you basically can see that these are your sequenced fragments, right? Now, each of those fragments contain these tags or these identifiers, and these are from your sense strand. And the antisense strand that has chip enriched fragments also, when they interact with each other, then what you would expect to see are that sense tags and antisense tags can be distinguished. So basically it shows you which way the gene is going to run. Next, you have to analyze how high quality your actual data is. So the QC of ChIP-seq samples is critical to judge whether sequencing data is of high quality or not, whether it can be used for further analysis or not, whether many different QC measures have been developed. Among them, you have two major parameters. The first is mapping ratio. If you look here, this is your first metric, which is going to show the read quality and what proportion of the sequenced reads have been derived from true genomic DNA. For example, the mapping ratio for samples that have been sequenced by Illumina HiSeq should be over 80%. The exception here is a sample for non-DNA binding proteins like IgG, which usually have a much lower mapping ratio. The second parameter for ChIP-seq data that needs to be considered is the depth of the read that has been generated as a result of sequencing. So read depth actually depends on how long your genome is and what is the signal to noise ratio. So what is the expected ideal situation? You should have ideally at least 10 million uniquely mapped reads as a minimum to analyze the sharp mode peaks of human samples. Broad histone marks have often have weaker signal to noise ratio. They require more reads as a practical minimum for peak calling. Library complexity, which is the portion of redundant reads, ranges from 0 to 1, and consortium suggests that complexity should be more than should be less than zero point 0.8. No, it should be more than 0.8 for 10 million mapped reads, and lower values indicate excessive PCR amplification from a small amount of initial DNA. Basically, what are we saying? We are saying what parameters need to be taken care of whenever you are uh, overall analyzing ChIP-seq data. First is the mapping ratio, which tells you the proportion of reads, what reads you have gotten. Among all of those reads, what percentage or what proportion has come actually from true genomic DNA and is not any other contamination. And second is the read depth, which shows you that among the reads that you have gotten, how much or what percentage or what proportion of them are non-redundant, which means they are unique, usable, uh, useful mapped reads. What percentage do they compose? So these are the two parameters which are rather important. Apart from that, there are a few others as well. The first is NSC, which is normalized strand coefficient, which is obtained by SSP, a signal to noise indicator for both sharp and broad marks. Um, when you carry out in-depth validation using more than 1,000 publicly available ChIP-seq data sets, you would see that recommended threshold is usually NSC more than 5, NSC more than 1.5. Input sample should have very low signal to noise ratio, and therefore NSC value should be less than 2.0. So basically, you do not have to remember all of this information, but you should get an idea whenever you wish to do ChIP-seq data analysis, what sort of parameters you should keep in mind. Then you, okay, there's a lot of noise. Then you have background uniformity. Sorry if there's disturbance, it's just the background. Then you have background uniformity, BU, which reflects the redistribution bias in background regions and which ranges from 0 to 1. So what is the implication of this particular parameter? When there's a low value of background uniformity, it suggests that the redistribution is more congregated or more biased, which gives you more false positive in the obtained peaks. For the genome, which bears extensive copy number variation, like for example, a breast cancer cell MCF7, a more relaxed threshold is is more desirable. So it shows you that of um, what is the bias in the regions from which reads have been generated, in the sense that reads which have been generated from background regions versus reads which are generated from your desired region. So again, just yet another parameter. Then you have GC summit bias, which reflects whether there is bias during IP step or when you have actually performed PCR to amplify your DNA. So generally the GC summit, which is similar to uh, percentage GC, which is considered during primer designing also. So in general, the GC summit of typical ChIP-seq data becomes similar to that of the reference genome. For reference genome, approximately 50% is the percentage GC for human beings, and unexpected GC rich summits PCR amplification biases, because sometimes the polymerases that you use for PCR amplification, they end up integrating random bases at the time of amplification, and that can lead to unwanted, unexpected, and random mutations. And false positive peaks can also be derived from certain regions which are hyperchippable. What does it mean, hyperchippable? Basically, these regions are very prone to getting immunoprecipitated. Basically, after fragmentation, there's a tendency of them to bind to the antibody of interest, and they are found as a common false positive or a common error during IP, or which is not IP, during ChIP. And yeah, it's a common error. Let me put it that way. Mhm. Then the next step comes normalization. So what do we mean by that? So basically, if you think about it stepwise, what have you done till now? You've actually done your immunoprecipitation. You've sent your samples for sequencing. You got your raw reads. You have pre-processed those reads, and then you have trimmed those reads and done the quality check. You have aligned it to the reference genome to know where that read has come from. Then you have checked whether the presence of that read is significant or not. So you have checked for peaks. When we say peaks, we basically mean that these regions have got precipitated multiple times from multiple samples, suggesting that these are regions which are commonly being um, regulated or commonly bound by the protein of our interest. Now, once that has been done, what did we do next? Let me just a second. Yeah. So after this peak calling step and after determining where exactly it has come from, then we have, or we can also do a ChIP-seq quality data quality assessment where we can check all of these different parameters, and then we proceed for further analysis. But again, it's not a very regularly followed step, but it's a good thing to keep in mind before proceeding too far. Then you have comparative analysis being the most relevant objective. Why do we say that? Basically, having an idea about whether or not a protein binds to a particular genomic locus is not always complete information by itself. What do I mean by that? Like, for example, if you have done ChIP-seq, all you will get to know is that at the point of time when you have harvested DNA from your cells, the chromatin from your cells, all you can infer from the ChIP-seq data of that cell is that at that point of time, under the control conditions, these are all of the genomic loci within the cell that are occupied by these number of proteins. That is all the information you'll get. So most commonly what is done is that apart from this control or wild type condition, there is some experimental condition, some variation where you kind of interpret or check for the difference between these two conditions, and not at a local scale but at a global scale, and it is essential to mitigate any technical variation before you can draw any conclusions. So for such comparative analysis, simple total read normalization very commonly is used, which basically sort of normalizes the read number between different samples to be the same. The, un the underlying assumption is that the difference in mapped reads among samples is sufficiently smaller than the total read number. What do we mean by that? The mapped reads among different samples is usually much, much less than the total reads that you have gotten from that particular sample. This assumption is not always satisfied, and therefore different methods have been developed which can identify differentially enriched regions between two conditions, some of which are designed specifically for histone modification data. So your idea is to check that between my control condition and my treated or modified condition, what is the alteration between these, not the systrome, but the changed binding of your preferred protein of interest to the region that you are studying. So that you want to compare, or you want to overall see the differential expression, differential enrichment of these loci among different samples. And since obtained results vary a lot among different tools due to underlying statistical assumptions, sometimes you can have a lot of variation among different samples. And so all of that also needs to be kept under consideration. Uh, Quantitative comparison between or across more than two groups is a lot more complicated. So when the expected signal to noise ratio is similar among different samples, you will see that for differential gene expression analysis, statistical methods can be used. It is also possible to utilize quantile normalization when the signal to noise ratio for most common peaks is similar. Basically, if you have multiple treatment and control conditions and you use the same antibody and the same experimental conditions among all of them, usually their sequencing data would be comparable between different ChIP-seq samples. If it varies a lot, then you have to do some calibration analysis. So these are all um, optimizations that need to be done whenever you are proceeding with the analysis yourself. I have kept this in case you are interested in studying it in more depth, but I'll not try to complicate things for you because this analysis itself is quite, it's not easy. It's a long process, but we'll stop it at here at this step, because the analysis optimization is endless. You can go to GitHub, you can go to you can mail the authors directly, you can look at commonly followed pipelines, and you can use Galaxy also. So these are all the different tools you can utilize, and you can optimize it based on your expected pattern or your hypothesis to get the most optimal results. So let us move forward. What you can also do eventually from your ChIP-seq data is carry out functional analysis, where you look for your motif of interest, so as to make sure that whichever, say for example, let me put it this way. Say for example, you found three regions within your gene of interest, one at the promoter, one within the gene body, and one downstream of your gene body. So there's one upstream, one downstream, and one within the gene body. These three sites have come out as significantly enriched peaks as the result of your ChIP-seq data analysis. Now, one way to sort of cross-check, validate, and functionally justify this enrichment is to check whether the consensus binding motif of your protein, which, if you remember, we in the previous class I had mentioned, you can find using tools like JASPAR, for example, or TransFAC, which is used by JASPAR. You can use that tool to screen through the entire region of chromatin, which is under your study, and you can check whether or not your binding motif is present in all of these sites. Then that allows more functional validation and more confidence in your data. So general motif analysis methods commonly have two types. You can either do novel motif discovery, which allows you to look for new binding motifs for unknown factors which are appearing in a large fraction of peaks, or you can do motif scanning, that is going to look for your sequence of interest and can rank the similarity of your supplied DNA sequence against known canonical motifs within a database. So ChIP-seq peaks are used for functional enrichment analysis. You can quantitatively rank nearby genes as potential targets, group them by gene ontology. You can do downstream pathway analysis. Opportunities are endless. But overall, what you want to get as an what you hope to get as an outcome of your analysis is prove whether or not your hypothesis is true, or check whether or not your hypothesis is true. So you will look specifically for your gene of interest. You will look for your downstream targets of your gene of interest, considering that it is a transcription factor. You can look at alteration in different pathways, overall pathways, if you have done some sort of a treatment or some sort of a disruption. So all of that information can be obtained. In addition to that, you can look at chromatin state annotation, also called the semi-automated genomic annotation, or SAGA, which classifies these genomic regions based on some patterns, whether it is a promoter, it's an enhancer, a transcribed region, a repressed region, using a machine learning approach. Again, this is an up-and-coming strategy. There are so many techniques. If you go about just look at ChIP-seq analysis in today's time, you will have so many different options, a lot of data that can be interpreted to give you a wide range of applications. So you can explore these opportunities if it is something of your interest. Most commonly, what people look for is whether or not it's an enhancer region that is getting enriched. So region-specific analysis usually involves enhancer analysis that narrows down the target genomic regions to be investigated. And chromatin state annotation basically segments the genome, assigns chromatin states to whole genome regions using an HMM, or hidden Markov model. And yeah, again, this is a little more complicated, not going into too much detail about it. But basically, you want to classify the region that you have found as an enriched locus of your genome into whether or not it is a promoter, an enhancer, or it has some distinctive property that is biologically relevant in the condition that you are studying. So yeah, so that was pretty much what I wanted to discuss regarding ChIP-seq data analysis. What I would recommend is for you to also go through this video. This is about how you can visualize this data. So what you get as an output usually is a bed file, which you can convert into a big file, and that you can visualize on a tool known as IGV. Maybe I'll just quickly go through the video as well before we take the discussion a little bit forward. So yeah, I think I had it open. Oh God. Let me check. I think my screen is dark, or is it not? Just give me a moment. No, I don't have it open. You can check it in your own time, but basically there are two tools that are most commonly used for visualizing ChIP-seq data. One is integrated genome browser and integrated genome viewer, IGV and IGB, which are most commonly used, where you can sort of upload your files and then you can visualize them. They sort of appear as tracks. Maybe I can show that to you in the next class. My browser is not working properly. So that allows you to visualize the results that you have gotten from your analysis. So that is the idea of ChIP-seq data analysis and the outcome that you expect to get from it. We will take up this discussion in the next class when we will discuss 3D chromatin architecture in a lot of detail. But basically, I just wanted to just introduce you to the concept that till now what we have thought about the interaction between any DNA binding protein and the DNA itself to be sort of two-dimensional in nature, where DNA can be thought of like a table and protein sort of like an object you have kept on the table. But inside the cell within the nucleus, that's not how it actually looks, right? Instead, it is sort of it's a three-dimensional. It's sort of a very looking very messy sort of a structure where you feel like there is no organization. But if you go into the depth of it, you'll see that it's so tightly regulated, controlled by n number of proteins, and very tightly controlling which region of the chromatin can be open, which cannot, and how much of it should be open for how long, and what sensor or what external stimulus is going to shut off that expression or can temporarily open up that expression. All of this is completely under control. So how that is actually brought about is by bringing about a new dimension to chromatin architecture, which is three-dimensional in nature. And in today's time, these techniques are becoming more relevant, more yes, definitely more complex, but also more informative in terms of how chromatin looks inside a cell in three dimensions. And there are different techniques which are used for studying that as well, which we'll take up in the upcoming classes. But just to introduce the concept of it, if you look at chromatin architecture, it has multiple aspects to it. You have cis-regulatory interactions. Just a second. Yeah. You have cis-regulatory interactions like enhancer-promoter interaction. You have repressive interactions like PRC group of proteins, which are polycomb group of repressive proteins and their interactions. You have LADs, which are lamina-associated domains. You have TADs mediated by different structural elements like cohesin, CTCF, etc. So if you look at the affiliation between 3D chromatin architecture and the hallmarks of cancer, you will find that it mediates the induction and repression of genes at multiple levels, like including the enhancer promoter looping, which is carried forward by transcription factors and remodelers of chromatin. So they impact all of the cancer hallmarks, which include sustained proliferative signaling, evasion of growth suppressors, resisting cell death, etc. You can find all of this information online or in my previous course that I taught last semester. You can read about the hallmarks of cancer there as well. But basically, the 3D chromatin architecture can influence all of the different parameters which are called as cancer hallmarks, or which are attributes that a cancer cell acquires over time, that or rather any cell acquires over time to become cancerous. So they are enhancing or they are aiding the oncogenicity of any cell, and therefore it is all they are all interconnected. And so studying such 3D chromatin architecture is relevant in that context. And also, apart from just oncogenicity, biological processes do get affected as well. So this is how chromatin looping, if you can resolve it to that extent, this is how chromatin looping can possibly look. So you have very distant loci between or very distant regions of the DNA coming into close contact of each other when they are bound by and controlled by different proteins. For example, if you look at this loop, it is allowing the interaction between two regions of the chromatin which are very far apart, and it is regulated by cohesin and CTCF. And here you can see the enhancer and promoter coming close together because of the cohesin CTCF interaction. And as a result, what it is doing is downstream of the promoter, whichever gene is present, that is getting upregulated. So 3D chromatin architecture has an immense role to play. We will discuss it in more detail in the next class. And it is one of the hallmarks of cancer, like I previously mentioned. And you can see that it can aid metastatic pancreatic cancer as well. And therefore, these proteins which help facilitate the formation of these 3D structures, they can be very commonly explored as therapeutic targets. And there are drugs which have been synthesized or which are under production under clinical trials to target such proteins and disrupt 3D chromatin architecture to make it a better strategy for cancer patients. And also it can influence cancer evolution and cell cycle, and therefore 3D chromatin architecture is very important, and we'll learn about it also. So that covers the discussion I wanted to do for today. So if you have any doubts, please let me know. We'll discuss those, and then I'll quickly brush up the topics that I want to cover for before our next exam. So just let me know if there's anything, because I know these are very new topics, so it might be a little loopy for you initially, but let me know if there's anything you want to discuss. Then we'll move ahead. No doubts. You could understand me. I thought it was a little
No, ma'am.
complicated.
Yes, ma'am, it is, it is.
Okay, anything you want to discuss, or like if there's anything you want me to elaborate more, maybe I can get more material for you in the next class. Because I didn't go into too much detail, but I felt like maybe you would need more information. It was a little brief, maybe. Tell me which portion you would like better discussed, and we can take it up in the next
Ma'am,
class.
the analysis portion, maybe it like
Mm-hmm.
actually it is very new to me, so maybe you can
Okay.
discuss that.
Okay, so I will go into, okay, I'll get some more material regarding the same, but just so that it's a little easier, let me find that slide. Where is it? Did you attend the previous analysis classes? Because the initial part of it is like quite similar, which is why I didn't go into that much detail. Like the alignment part, this part you must have attended. I remember a lot of this discussion went on for quite some
Yes,
time.
yes, yes, ma'am.
Yeah. So after that, when we actually began the analysis, we had initially started off this discussion only. Mm, yeah. So we had discussed data formats, and then we had discussed quality check using FastQC and Trimomatic. So that part is pretty much the same. So whenever you have a raw read, this initial part is done. Like this is a most obvious step that you need to follow. So once that is done, the next part I believe is what is more complicated. So this is the Trimomatic part. I had discussed it in detail back then. And after that, yeah, then I had introduced the reference genome, and we had a little bit of a discussion on the Human Genome Project and its relevance. And then we moved on to alignment. So maybe I'll elaborate a little bit more from this point onwards. So uh, the tools we had discussed previously were STAR, which is more commonly used for RNA-seq data analysis. And different sub-steps in this process include indexing the genome, then aligning it. Like you break down the entire genome into little small fragments, and you index which region corresponds to which area of the genome, and then you do the analysis. So you don't do all of this. All of this is done by the software, but it is the code for it is what instructs the software how to proceed. So maybe I'll go from that point onwards. We had discussed global alignment and local alignment, and the different strategies which are used by different tools. Star aligner particularly was discussed in more detail. So anyway, don't worry, I'll discuss it with you. And the difference also between these tools lies in the fact that for star aligner, a separate consideration had to be kept, which is that when you are doing RNA-seq data analysis, you have to make sure, or the tool has to make sure that when it is aligned to the reference genome, it is taking into consideration the fact that your RNA is not going to have exons versus the DNA, of course, is going to retain exons. So it should be able to map accordingly. So all that is not a headache when you are carrying out ChIP-seq data analysis. So you don't need the star aligner. You can use it after modifying certain parameters, but Bowtie2 is the more preferred tool in that context. So yeah, this was all part of the way in which this tool works. And as a result of the alignment, the data format you get was the SAM format, which is then converted into BAM format. Again, what I told you today, which is a binary form of the same thing, which sort of constricts the amount of data you have. Because SAM, usually each file is of 1, 2, or 3 GB versus BAM is around 200 or 300 MB, or sometimes much less than that also. So it reduces the processing power which is needed to handle that sort of data. And yeah, that is pretty much where we stopped our discussion. But okay, don't worry, we will take forward this discussion in the next class. So maybe on Friday, I'll cover that discussion before moving on to 3D chromatin architecture. We can keep that for next week. So, anything else you want discussed? Because I will brush up the topics for another half an hour or so, and yeah, then we end up today's class. Anything you want to discuss? Any topic that till now that you need to be discussed in more detail before we move on to 3D chromatin architecture? You can let me know. Um, Yeah, so this is where we had paused our discussion in our, I think in the first test that we had for Sanger sequencing was the last topic that we had covered. So that point onwards, I want to carry out the discussion for the next exam. So if you remember, we had begun our discussion on next generation sequencing techniques. So from this point, NGS techniques, introduction, the template amplification strategies, sequencing by synthesis, short read, long read sequencing methods, and this initial discussion on exome and WES. I want to keep it till here. So slide number 74 would be the last slide that I would like to cover for the next exam. So you will have somewhere around 30-35 slides only, I presume. So that will be pretty much, yeah, less than that, much less than that. So that will be the syllabus that I want to cover. So this portion is what I'll revise in today's class briefly, and in the next class as well. If you have any doubts in that region, please let me know. And then we'll have the exam then on 16th March, 16th or 17th March, whichever is feasible, possibly on 17th, that is Tuesday. So please let me know if there is any confusion, any doubt you want clarified before that, so that I can sort of help you cover that before the exam can be executed. Um, okay, if you don't have any queries, I will begin the revision from this part. So basically, our discussion began with understanding of next generation sequencing methods. So what do you exactly mean by that? It is a high throughput sequencing method. What does it give you? It gives you A, lot of data, B, gives you scalability, and C, can take care of the speed. So it can either give you two types of data can be obtained from high throughput sequencing. Either it can be a lot of information which is corresponding to the same input. Like for example, what we see for short read sequencing, where you have short fragments which are going to correspond to the same region. Or it can give you long read sequencing data where you have long reads for the same region, and both of them are used for different purposes. Again, we will quickly revisit that as well. But effectively, what these sequencing results give you are the order or the arrangement of nucleotides in the genome or in the RNA whichever is the input sample that you have sent for sequencing. So what does it do? It allows us to just a second. It allows us to rapidly sequence entire genomes. It can allow us to sequence with a lot of depth, a very small target regions. You can use RNA-seq as well to discover whether there are variants of RNA, whether there are splice sites, whether there is an alteration in RNA expression under a certain condition, whether there can be genome by DNA methylation, DNA protein interactions like we saw for ChIP-seq, chromatin accessibility, which we will cover pretty soon. It allows you to sequence entire patient samples as well to study different, study tumor evolution, study tumor subclones, somatic variants, whether there is a genetic basis to any disease or not. All of this can be done. So broadly, these are the different applications of any NGS method. But if you look here, you'll see that largely the difference between these two techniques is quite stark. In case of Sanger sequencing, you will have one single template that undergoes multiple rounds of sequencing. You carry out extension, arrangement, and then detect the order of the sequences. Versus in NGS, you'll have millions and millions of templates, which are the result of your signal amplification strategies using PCR and different methods, which are then arranged on a flow cell, which is sort of like a slide. Imagine a slide or a chip onto which these can be arranged. And then extension via PCR is carried out, and then you detect which base gets incorporated at each position, and that you detect using a laser, which is able to identify which fluorochrome just got integrated, and that gives you the effectively gives you the sequence of the region that you are studying. Please interrupt me if there's any doubt. Please do not shy. It's completely fine, and we are reaching the end of the course, so it's a good idea to clarify any queries you might have, so that I can also do a better job and explain it to you a lot better. Okay. So, what is the overall flow? What entails the overall flow of next generation sequencing? So, if you look here, you'll see that um, there are some common steps that you'll usually find. The first most common step is that DNA needs to be fragmented. The question is why? Why can't we just take long stretches of DNA and just sequence it? A, there's no enzyme that has such high fidelity. B, it's going to be erroneous. C, it can pose steric hindrance. And D, you do not want any bias in any region. So shorter regions are going to give you a more uniform template that is available within the reaction setup. And therefore, there is no preferential or no bias basically at the time of sequencing. So workflow of conventional versus second generation sequencing. So with high throughput shotgun Sanger sequencing, what does this mean? This shotgun term, if you remember during our initial classes, I had discussed that shotgun literally means like, for example, if you shoot something and as a result of it, the explosion gives you shorter sequences. This is pretty much what this fragmentation is also looking like. It is just going to give you smaller pieces of your entire genome. So genomic DNA is fragmented, cloned into plasmids, and used to transform E. coli. Now, for each sequencing reaction, one bacterial colony is picked up, plasmid DNA is isolated, and in each cycle, sequencing reaction takes place within a microliter scale volume, generating a ladder of DDNTP terminated dye-labeled products. So if you remember, this is what we had seen. It pretty much looks like you are going to get um, end-terminated or chain terminating sequences, the concept of Sanger sequencing that we have already discussed, because a dideoxynucleotide phosphate is incorporated instead of a deoxynucleotide triphosphate. So wherever a DDNTP is inserted, there the sequencing reaction stops, and the order in which these ending of the sequence can be seen, you can detect the sequence of the overall template in that manner. Again, to repeat what I just said, this is your initial template. You break down the template into smaller fragments. You transform it into bacteria. Bacteria are able to grow and form individual colonies. You pick each colony, and then from each of these colonies, you carry out a sequencing reaction. Within very few microliters, you will get a sequencing reaction performed. Now, as a result of it, what you'll get are these differently labeled fragments. What has the label been decided by? The label is decided by the DDNTPs which are present in the reaction mixture, where each color shows you each different fluorochromes that can be recognized by the excitation of a by exciting them with a laser. So these fluorochromes are going to indicate which base is present there. If there's yellow, it shows C. And after C, the next length shows you, or the next base will be extended by a certain unit that shows you a blue fluorochrome. So the next base is T, the next base is A, and so on and so forth. So that is what is done in a high throughput shotgun Sanger sequencing. Um, so yeah, so this gives you a sequencing trait. Then you trace. Then you have shotgun sequencing with cyclic array methods whereby common adapters need to be used. So after you have fragmented your DNA, you attach these tiny sort of light blue colored adapters, which is shown here in light blue. You attach them to the end of your DNA fragment, and then it is subjected to one of the several protocols that results in the generation of polonies, which are PCR colonies that are spatially immobilized, and each polony has many copies of the single shotgun fragment because it has divided in a clonal manner. Now, as the polonies are tethered to a planar array, a single microliter scale reagent volume can be applied to manipulate all of these parameters. And when multiple times you do these enzymatic interrogation, you basically do multiple rounds of sequencing where you have more than 10 to the power 6 reads per array, you'll be able to detect which which base is present at which location, and from there sequencing can be done. Now, as you can see, these techniques, while are based on somewhat based on high throughput sequencing and can seem promising, they also have their limitations, which has brought us to third generation sequencing eventually, which is more advanced and more uh, technically more reliable. Now, second generation usually refers to various implementations of the cyclic array sequencing method, which I just discussed, that has been realized in a commercial product like 454 pyrosequencing, Solexa, Solid Platform, Polinator, etc. These are all commercial machines or commercial setups. And the concept of cyclic array sequencing can be summarized as the sequencing of a dense array of DNA features by multiple repetitive cycles of enzymatic manipulation. You manipulate the enzyme and you capture the data using imaging strategies. So that is cyclic array sequencing. Again, it's not the most common strategy used today, but it is something that has been done previously. Now, beyond that, for sequencing, what is the practical way or in which this is carried out? You have the 454 pollinator, like I just mentioned here, the 454, the pollinator, solid sequencing platform. They all rely on emulsion PCR. What is emulsion PCR? Simply put, within a small droplet or within an emulsion droplet, the PCR reaction is carried out. So what does it do? It amplifies clonal sequencing features. So briefly, an in vitro constructed adapter flank shotgun library. If you remember previously, we saw that the shotgun library was labeled with adapters. Here the adapter is shown in gold and turquoise, which flanks your insert. That undergoes PCR amplification. This PCR is multi-template PCR, not multiplex PCR, because a single primer pair is used in a water and oil emulsion. Now, one of the PCR primers gets tethered to the bead that is also utilized in this reaction. This is the bead that you can see. The bead has your turquoise colored adapter or primer, turquoise colored primer present or immobilized, to which this adapter sequence will be able to bind. And a very low template concentration is usually utilized. And then from here, PCR can be initiated when it is within the emulsion. As you can see, within your emulsion droplet, you have your beads, microbeads with the immobilized adapter, and you have your sequencing fragment that has adapters attached on either end. Now, apart from that, you also have DNTPs, you also have the enzyme that can actually execute the reaction. And what it basically does, what is the point of doing this emulsion strategy, is that it basically constricts the area within which PCR amplification can take place, which increases the efficiency, reduces the time, and makes this strategy quite effective. So yeah. So PCR products or PCR amplicons are captured on the bead surface, and then they can be selectively enriched. The beads can be enriched, and on its surface, what you will have is the PCR product, which you can purify. You can see you have gotten the sequence for, and you can purify also if you want to use it for other purposes. So what this entire strategy is basically doing is you are increasing the amount of template by polymerase chain reaction. So more and more template can be generated for the actual sequencing step. The other strategy, the one we actually discussed in a lot more detail, which is more extensively studied and used, possibly because it is run by Illumina, which is the Celexa technology that relies on something known as bridge PCR or bridge amplification, which generates clusters. As you can see, there are these distinct clusters of same insert here, one in blue, the next one in sort of this orangish brown color, that is generated in sort of in local areas of concentrated enriched template. So briefly, an in vitro constructed adapter flanked shotgun library undergoes PCR amplification, and then both of the primers are able to coat the surface of a solid substrate. This is your solid substrate. Think of it as a flow cell or a chip, on the surface of which you have primers present or sort of immobilized on the surface. They are attached at their 5' ends by a flexible linker. If you sort of zoom in into this region, you will kind of be able to see this immobilization strategy as well. As a consequence, amplification products which are originating from any given member of the template library will stay locally tethered very close to the surface. Once PCR is complete, each cluster will contain around 1,000 copies of each member of the template library. So imagine how much amplification has occurred because of this strategy. Accurate measurement of the concentration of the template library is also very critical to maximize the cluster density while avoiding unnecessary overcrowding. So this is a part of the library preparation step where you have random fragmentation of DNA, you have in vitro ligation of your adapters, and then you generate these locally clustered amplicons, which can be used for sequencing strategies. The details of this protocol you can find on this link. We had discussed this in more detail. You can also refer to this class. I think it was the 5th or 6th class possibly. Maybe I can check here. Okay, we'll come to it later. But basically, this is the chemistry behind it, how this bridge amplification thing works. So when it came about, it was a sort of a revolutionary step in science because it's quite unconventional. What it does is it uses alternating cycles of extending with the polymerase known as the BST polymerase, followed by denaturation with formamide. So what does it do? It is basically, if you look here, this is what it looks like. You have your primers and you have your adapter sequences as well. You add nucleotides and polymerase, and from that point onwards, extension will begin. You image the array. Once a single base has been incorporated, it stops or it quenches the reaction. It takes a snap and it images the array, removes the label and the terminator. Then the reaction can proceed again. Again, you take an image, it continues, and this is sort of repeated in a loop. So after cluster generation, amplicons are single-stranded or linearized, and a sequencing primer is hybridized to the universal sequence, flanking the region of interest. Now, each cycle of sequence interrogation consisted of a single base extension with a modified DNA polymerase. Here, that is known as the BST polymerase. And yeah, the BST polymerase. And along with that, you have a mixture of the 4 nucleotides, which are going to allow the continuation of the PCR reaction. The nucleotides are also modified. What is modification? First, they have reversible terminators. What does it mean? It is a chemical substituent group at the 3' OH position. What is that going to do? If you remember Sanger sequencing, there we had a DDNTP, right? So the DDNTP is not allowing the reaction to proceed once that base has gotten incorporated. Here, what is present? The 3' OH group modification has a chemically cleavable moiety, which is allowing only one base incorporation to occur, and only one of the four fluorescent labels, which are also chemically cleavable, will correspond to which base has gotten incorporated. Now, after one base extension has occurred, image acquisition has been carried out, both the groups are cleaved. And it is now the entire template is extended by one base and is ready for the next round of incorporation. So that is pretty much what you saw here. So this continues over and over and over again, and then the bridge folds back on itself. The unattached the unattached DNA fragment is washed off, and the next DNA fragment is linearized to allow another round of extension to proceed. Again, I know just hearing it might be a little confusing, so it is a good idea for you to go back to that official video by Illumina, because they have a beautiful animation of how this strategy works, and it's a good idea to learn it because it's an indispensable technique that is so revolutionary and such an unconventional, such a unique idea to study. So it's a good idea to learn about it. Okay. Next, this is the official introduction into template amplification strategies. The first one is emulsion PCR, like I just mentioned, which is followed by all of these different companies. First, you have formation of an emulsion, where you have a water in oil emulsion, and within these you have your bead, you have immobilized adapter sequences or primer sequences, and you have your adapter bound template. And on bead amplification is carried out where the templates are able to bind to the primers, and then extension can take place. And the final product has 100 to 200 million beads, which have thousands of bound template. The second strategy, which I just mentioned, is the solid phase bridge amplification strategy, where you have this sort of flow cell on which you have um, templates which are hybridized that are able to interact with near primers where amplification can take place. And as you can see, this sort of bridge formation occurs, which allows the completion of the complete strand. And generation of clusters occurs when there are many rounds of amplification. Nearly 100 to 200 million clonal clusters can be formed, which leads to immense amount of cluster amplification or template amplification, which makes which can enhance the depth of your sequencing as well. And this is done within, like, if you zoom in, this is what the pattern flow cell looks like, and that has these sort of micro wells sort of structure, which direct cluster generation and increases the density of each cluster, which can make your sequencing report more reliable and have more depth. So apart from that, there are also these two other strategies. The first one is the solid phase template walking strategy, which is used by solid for and what they do basically is a little bit similar to bridge amplification. But yeah, instead of the formation of that bridge that we saw here, it's more sort of like a immobilized surface onto which you have DNA templates, onto which you have DNA primers, to which your template is able to bind. And then from this point onwards, you can see synthesis takes place. Then you have primer walking, where there is partial denaturation of this DNA, as a result of which the free end of the DNA can hybridize to a nearby primer. Then there is again regeneration of template from that point, and this keeps on continuing over multiple rounds, as a result of which you will have again clusters of DNA, similar to what Illumina was also doing. But yeah, the difference being the formation of that bridge where the DNA is bending backwards over itself. Then the fourth strategy, which you will more commonly find for long read sequencing, is an in-solution DNA nanoball generation, which is taken care of by companies like BGI Directed Complete Genomics. So there you have your template bound to adapters. Then one set of adapters is ligated to either end, followed by circularization of the template to generate the covalently closed DNA molecule. Then circular DNA templates are cleaved downstream of the adapter sequence. And after cleavage, what is done is you add another round or another adapter, again recircularize it. You do it again, and you do this 3 to 4 times, where you have multiple rounds of iterative ligation, where you have ligation, circularization, cleavage, again ligation, again circularization, again cleavage, so that you have 4 different templates here shown in red, blue, green, and yellow. And what happens after that is the most defining step of this technique, that is the rolling circle mode of amplification. What happens there? If you have learned about the rolling circle mode of DNA replication, then you know that it generates concatenated sequences, right? So here you have circular templates which are generating long concatemers called as DNA nanoballs. And then these nanoballs undergo the sequencing step that we are interested in. And the nanoballs are cohesive and separate in solution because of intermolecular interactions. They do not loop against or loop among each other. And after that, DNA nanoballs can be immobilized on a pattern flow cell, like you can see here, and then that undergoes sequencing. So this library molecules undergo a rolling circle amplification step, generating these concatenameric masses, which are deposited on a flow cell. Maybe I will pause here. Yeah, because this was only the sequencing by synthesis method. Yeah. So from sequencing by ligation method, I'll revise in the next class. But if you had any doubts till this point, please let me know. I'll discuss this, and then we'll wrap up today's class. Please let me know if there are any queries, anything you want discussed, or anything basically you want to say.
Ma'am, it is clear to me.
Okay. So this part you understood, right? Because I think this was discussed also in
Yes,
more
ma'am.
detail. Okay, okay.
Yes,
Okay,
ma'am.
I'll go into details about the chip-seq analysis also, so that it's clear for everybody. Uh, anything, Mayank, anything you want to discuss? If not, then I'll wrap up today's class. Okay, I don't know if it's here or not. Okay, anyway, thank you for joining. We'll have the next class on Friday, that is on 13th, and we'll discuss chip-seq analysis data in more detail, and I'll also revise the remaining topics for the test that we'll have on Tuesday. So the next 3 weeks, I think we have 6 or 7 classes left. I think 6 classes left and one exam. So these classes will have twice a week so that we can wrap up in time. So next week we'll have 17th and 20th, 24th, 27th, 31st, and 3rd. And that should pretty much cover whatever remaining topics we have within the previously mentioned duration of classes. So anything you want apart from what I've already taught, if there's anything else also that you need for your own courses or own careers or whatever you are interested in, you can let me know, and we can have those discussions as well. So thank you so much for joining. I'll see you again on Friday, and we'll keep the classes from 6:30 so that we can wrap up in time. But yeah, thank you so much for joining, and have a good night.
Good evening, ma'am.
Yeah. Hi, Mayank. You had to say anything?
Yes, ma'am. I will hear. Man, I will take all class, but at that time, I am preparing for my competitive exams.
Yes, yes, I received your email. Yeah, I understand.
Yes.
It's okay. I hope it went well. Most of the
Uh,
topics have been uploaded. Like I think everything is uploaded till before ChipSeek. So we have covered very wide range of topics. So you can learn
yes.
it in your own time. Don't worry. The initial topics are quite basic. They were core molecular biology. Um, then we moved on to the techniques and the analysis steps, and that came on later. Some of it I'll already revise, but don't worry, just try out the test yourself. It's more than anything, it'll be for your practice. So it's a good idea. I think some people, even if they didn't attend the test live, they tried it out later on and they scored really well. So it's okay. You can do it for your own learning. Don't worry about it. The analysis part is a little complicated because I'm not doing it live with you because I don't have all of that installed in my system. We do it in a common Linux system here. So that becomes a bit of a problem, but at least I want you to learn the theory of it and the basics of it so that you can do it whenever you are interested. So yeah, so that's the plan. But anyway, I hope you'll attend the upcoming classes and I'll see you both again. So okay, thank you. Good night.
Thank you,
Thank
ma'am.
you, ma'am.